This video looks into feedback circuits in the digestive system and how they regulate digestion, energy storage, and appetite. In animals, digestion is activated stepwise, meaning that each process of digestion starts when it receives a signal from the process before. All processes in digestion are regulated by the enteric nervous system, which corresponds to a network of neurons that together with other systems ensures the correct functioning of the digestive system. The endocrine system also plays an important role in regulating digestion, specifically in the stomach and duodenum, the first segment of the small intestine. This regulation is known as the hormonal control of digestion. For example, when food is eaten, the stretch of the stomach is recognized by neurons, which send signals to release gastrin. These hormones stimulate the production of gastric juices. Another example is the work by cholecystokinin, or CCK, in stimulating the release of digestive enzymes in bile when chyme passes from the stomach to the intestine. Secreting also stimulates the release of bicarbonate to neutralize the acidity of chyme. Secreting and CCK also have an opposite effect when the chyme is rich in fats. Here, both hormones inhibit peristalsis and the release of gastric juices to slow digestion. It is also important for animals to regulate energy storage as glycogen. In humans, glycogen is stored in the liver and in muscle cells. However, if there is excess energy, this can be stored as fat in adipose cells or adipocytes. When talking about energy, we need to consider glucose and how its levels are controlled in the blood. In humans, the normal glucose concentration ranges from 70 to 100 milligrams per 100 milliliters of blood. When levels drop or become too high, the pancreas releases either insulin, produced by beta cells, or glucagon, produced by alpha cells. First, let's consider a scenario where glucose levels rise. Beta cells in the pancreas will secrete insulin into the blood. Insulin will stimulate different cells so that they take up glucose and stimulate the liver to store glucose as glycogen. On the other hand, when levels of glucose drop, alpha cells secrete glucagon, which promotes the breakdown of glycogen in the liver and the release of glucose into the bloodstream. In normal conditions, as we saw, if glucose levels rise, insulin will stimulate cells to take glucose and store it as glycogen. However, sometimes glucose homeostasis is disrupted, often resulting in critical health conditions, like the famous diabetes. Diabetes mellitus corresponds to a deficiency of insulin or a decreased response to insulin by target cells. If cells are unable to take up glucose, fat becomes the main source of energy. However, acid metabolites are produced during fat breakdown and they accumulate in blood. The low pH in the blood and depletion of sodium and potassium ions results life-threatening. Diabetes occurs in two types, 1 and 2. Type 1 diabetes is insulin dependent, as beta cells in charge of producing insulin are destroyed by an autoimmune response. Patients with this type of diabetes require insulin injections to cope with the lack of insulin. Type 2 diabetes is non-insulin dependent, so insulin is present but target cells do not respond properly. This reduces glucose take-up by cells, increasing the glucose concentration in the blood. This condition is hereditary, or it can also be developed in people with overweight or due to the lack of exercise. Now, think about what will happen if your body could not recognize how much food you eat. This will lead to overnourishment and severe fat accumulation. To regulate appetite and consumption, a network of neurons target a region in the brain called the satiety center. In turn, certain hormones are secreted to regulate short and long-term appetite. One of the hormones involved in these processes is ghrelin, which stimulates hunger before meals. Interestingly, if you get on a diet, ghrelin levels increase, making it more difficult not to eat. Insulin and PYY, on the other hand, suppress hunger. As we know, insulin will act by lowering glucose levels and PYY will counteract ghrelin. Leptin, a hormone secreted by adipose tissue, also suppresses hunger when body fat decreases. If fat decreases, leptin levels decrease and appetite will increase. I hope you found this video really helpful. The concepts and information presented in this video will be true no matter what biology class you are taking. However, the concepts presented in this video are referencing material currently covered in Baylor University's coursework. Images and diagrams are from Campbell's Biology 11th edition unless otherwise stated. Remember, if you are a currently enrolled Baylor student, we offer free tutoring services in our tutoring center, which is located on the first floor of the Sid Richardson building. You will find all of the details you need to know about these services in our website, which is www.baylor.edu/tutoring. You can schedule a free 30-minute one-on-one tutoring session online or just drop in during any of our business hours. For any information about our current services, please visit our website. Thank you.